Hello, my name is Steven Rehen. I'm here from Brazil, and it's a pleasure to be here sharing some of our data in this thermal event. So here we are working in Rio de Janeiro with iPS cells, and we are using those cells for different projects. The lab is more known for the work with the Zika virus. And we used this kind of approach with induced pluripotent stem cells and neurospheres and brain organoids to try to understand how the virus infected the developing brain tissue. But besides this approach, we are also very interested to use these cells to understand other phenomena associated with brain function. Our lab, I'm showing now in this slide some of our collaborators in this project and then other projects of the lab, and also the sponsors of the work that we are doing in Brazil. As I mentioned, we are very interested to understand how the brain works, and to do that, we use iPS cells. In this slide, you are seeing nerve stem cells derived from iPS cells, and this is our main source of cells that we use to differentiate them in different cell types observed and present in the brain. This is one way that we believe it's important to complement some of the work that is done using postmortem brain tissue, mouse models, neuroimaging, and other approaches. So when you differentiate neurons or astrocytes or nerve stem cells from iPS cells, we have a very interesting tool to work with models of the brain. And it becomes much more interesting after the very important work of Shinya Yamanaka, reprogramming cells or creating pluripotent stem cells from, for example, fibroblasts. In the lab, besides using these induced pluripotent stem cells derived from fibroblasts, we also use cells that we obtain from the urine of patients. So because of this approach, we were able to obtain cells from patients, very ill patients, for example, with Alzheimer's disease or mental disorders, or patients with disease associated with development. And we have now a bank with cells from different kinds of mental disorders, and these cells are available for scientists interested to work in the field. So I really welcome all of the scientists that are interested to use iPS cells derived from patients with some mental and neurological disorders that are available in our lab. So with this kind of tools, right, like human nerve stem cells, astrocytes, and neurons, we are working with different questions to try to understand and to try to explain that happens in the brain. And we are also working with brain organoids. It's a very interesting way to culture induced pluripotent stem cells and to understand and to study a little bit better some of the aspects of the network of cells within the brain. One thing that is very interesting, and for me and for other guys, it's also become a very challenging kind of question to study, and a very exciting kind of challenge, I would say, is the work with the psychedelics. Like if you see in this slide, until the 70s, there was a pretty interesting amount of studies about the potential or therapeutic potential of some of these psychedelic compounds. Then, because of the war on drugs and other questions, political questions especially, there was a very decrease in the number of studies using psychedelics. And now, more recently, like in the last 10 years, there was what we call psychedelic renaissance in the sciences. Because of different scientists, for example, in Europe, in the US, and also in Brazil, they are starting to study again these compounds. In Brazil, we study different compounds, different psychedelic compounds, but especially One that is associated with some of our culture environment. And one of those compounds came from one mixture that we call ayahuasca. The ayahuasca is being used for several years from the shamanic and Indian populations in the country, and also in the whole South America. And recently, some scientists from the northeast of Brazil, especially Braulio Araújo and Siddhartha Ribeiro, they are testing the potential of ayahuasca as an antidepressant drug, more specifically the compounds that are present in this ayahuasca that's associated with some religions in Brazil. For example, Santo Daime, Barquinha, and UDV are examples of these religions that are developed in the forest areas of our country. And when you look at this ayahuasca compound, it's a mixture of two different plants, and it's possible to identify the main compounds present in this mixture. So in this slide, you are seeing examples of compounds present in ayahuasca. One of them is called armine, and the other is called dimethyltryptamine, or DMT. That can be present in two different forms, N-DMT and 5-male-DMT. N-DMT is the mainly DMT present in the ayahuasca compound. And they have this kind of hallucinogenic characteristic, right? So they are molecules that are involved in altered states of consciousness, such as dreams, free imagination, and insightful creativity. So this is what's believed that's how DMT creates these psychedelic effects in the brain. But our mind seems very important for these DMTs to reach the brain, because during the rituals, the people, they drink this tea, this mixture that we call ayahuasca, and then if there was no armine in this mixture, the DMT was going to be inactivated by the liver monoamine oxidase, the enzyme, right? And then the effect was not the DMT was not able to reach the brain, without causing the effect. So this was the mainly believed role of armine in this mixture. But we decided to see if armine would be able to have any direct effect in the brain or in cells from the brain. So in order to do that, we started testing the effect of armine in neurostem cells derived from iPS cells. In this slide, you are seeing the approach that we used, the workflow that we used for the first tests. So basically, we plated the neurostem cells, and then we exposed them to armine. And after a different period of times, we tested for proliferation, increasing proliferation, self-viability, differentiation, etc. And what we saw that was interesting is that there is an increase in the number of proliferating cells, the number of neurostem cells after the exposure to armine. It's interesting, and not only the proliferation of these neurostem cells, but also from radioglial cells. And this, for us, was a very interesting result. This armine was not able to cause DNA damage or cell death in our cultures, but also is something important to be studied. The armine is able to inhibit the monoamine oxidase, but also another enzyme called DIRK1A. So we decided to test which would be the preferential pathway on how armine would increase proliferation in these neurostem cells. So we compare the effect of armine with two other compounds, one that's a well-known inhibitor of DIRK1A, and the other is an inhibitor of monoamine oxidase. And what we saw is that the amine-mediated inhibition of DYRK1A is probably the mechanism or the signaling pathway that increases neurogenesis in vitro. This work was published, and because of this first work, we decided to explore other compounds present in the, not only in the ayahuasca, but also in other psychedelic substances. What was interesting also for us, because of this result, is the fact that DYRK1A, this kinase, is also associated with Down syndrome and is also considered as a risk factor for Alzheimer's disease. Moreover, it was published recently also that DYRK1 increases the proliferation of human pancreatic beta cells. So we believe that there is very interesting biology in the study of our mind. But let's return to the other compounds present in these psychedelics. As I mentioned to you in the beginning of the presentation, DMT, this compound with this hallucinogenic effect and the psychedelic effect in the brain. So, and you have this DMT in two forms. One is NN-DMT, and the other is 5-meo-DMT. We decided to expose ourselves to 5-meo-DMT that's present mainly in this, also associated with some ritualistic manifestations in Mexico, for example. So we expose our nerve stem cells to 5-meo-DMT, and we didn't find any result, or in other words, we didn't find any change in proliferation of those cells when they are isolated in vitro. But we observed also that, like, these cells in this stage of development, they don't have the serotonergic receptors, and you can see here this result in the letter A in this slide. So they have the sigma 1 receptor, but they don't have the serotonergic receptor that they are activated by DMT. So we moved on to another model, right, that I also introduced in my presentation, that these are the human brain organoids. And before we did these experiments, we confirmed that, like, these cells within the brain organoids, they have the 5-meo, the sigma 1 receptor, and also the serotonergic receptors, as we can see here in figure H. So we expose these brain organoids to DMT, and after 24 hours, we prepared the material to do proteomics, shotgun proteomics. And it was a very interesting approach. We did it in collaboration with other colleagues from in this case, from São Paulo, Daniel Martins and others. And it is a very an elegant approach to see what would be the changes present in the tissue after being exposed to DMT. We published this paper, and it's a very interesting, and for us it's an honor to say that, like, this paper became one of the top 100. Actually, it was among top 25 papers published in this journal last year among 25,000 papers. So for us, it was very, very interesting contribution to the psychedelic science. And what we saw with this data. First, we were able to identify several proteins, almost 100 proteins, changed by the exposure, by when cells in the human brains, in the human brain organoids, they were exposed to DMT, right? And more than 300 of them were downregulated, and almost 600 were upregulated. And with this information in hand, and with the collaboration from our colleagues, we decided to see, using some algorithms and softwares, what would be the pathways changed by within the cells when exposed to DMT. So in this slide, you're saying for example, inhibition of inflammation in the neural tissue when the cells are exposed to 5-meo-DMT. We also observed that like pathways associated with neurodegeneration and cell death, they were also inhibited by 5-meo-DMT, which we believe is interesting as to be explored as a potential therapeutic effects of this substance. So the goal of this work was basically to highlight and to identify pathways changed by DMT in the brain tissue. Besides this change, we also observed a reorganization of cytoskeleton and a change in the dendritic spine. We also observed that like the long-term potentiation components are modulated by 5-meo-DMT, which would suggest that like some mechanisms associated with plasticity could be altered by DMT, highlighting the importance to study psychedelics, maybe with the potential to become new drugs to treat, for example, depression. So what we basically did was to try to explore the cellular and molecular aspects of this psychedelic and to contribute with the other studies that are applying it, for example, in the context of the ayahuasca for depression in patients. So we believe that our contribution could help to better design new clinical trials using, for example, our mind or DMT. So this slide basically summarizes what I showed to you. That would be one possible effect of our mind in neurogenesis. We, of course, observed it in vitro, but we believe, and there are other colleagues trying to explore this aspect, especially in this case in vivo, and other colleagues are doing that, to explore the potential of our mind inducing neurogenesis. And because of the already described effect of ayahuasca in depression, we believe that maybe this is the molecular explanation for that. And the other compound that we studied in this, and that showed you in this presentation, is DMT, which, as I mentioned to you, is associated, at least in our model, in the signaling pathways that are connected, for example, with brain plasticity and with neuroinflammation. In summary, we show that like our mind enhances neurogenesis in vitro through the inhibition of DYRK1A. We also show that there is a change in the global protein expression after exposure to 5-meo-DMT, and we highlighted changes in the anti-inflammatory signaling pathway, and also proteins involved with learning and memory. And we also observed a downregulation of the mGlu-R5 after the exposure of 5-meo-DMT. And it's also interesting since there are also some scientists working on the potential of 5-meo-DMT as a way to treat drug abuse. And finally, we observed that there is some cell death pathways inhibited by 5-meo-DMT. So there are interesting biology and potential therapeutic approach or applications for psychedelics. And this is just examples of two of them that we are studying using induced pluripotent stem cells, neurospheres, brain organoids, and the cell types that we're able to raise in our cultures. Thank you very much for your attention, and it's always my pleasure to work with Turbo and to share with you the kind of work that we developed here in Brazil, especially in Rio de Janeiro, in the IDOR Institute for Research and Education, and also at the Federal University of Rio de Janeiro. And in this slide, you have the website from our institute and also the website from my lab. Thank you very much.